So now that we've talked about amino acids and how they join together, we can start talking about the complicated structure of proteins. This is usually something that scientists cannot predict, and they actually use X-ray crystallography to determine it. There's many different intermediate structures that proteins go through on their way to their final stable shape. We're going to talk more about that when we review cells, and we're going to talk about how proteins are translated, but in X-ray crystallography, what actually happens are X-ray beams are shot toward a protein, and how they diffract or how they bounce off the molecule and is how scientists determine their overall shape. So it's a complicated process. So proteins are very, very complicated because they have lots and lots of different structures, and that means that they have the most varied function in living cells. So proteins are by far my favorite biological molecule, but because they do the most work, like they have lots of different functions. And what that means is the more functions you have, the more variability you have in your shape. So when we're going to talk about the structure of proteins, there's going to be 4 levels of structure that we're going to talk about. The first we're going to talk about is primary structure, just a sequence of amino acids. And then we're going to talk about the folding that's created in secondary structure. And then we'll talk about tertiary structure. All polypeptides and all proteins will have primary, secondary, and tertiary structure. However, a 4th level, quaternary structure, that only happens when I have multiple polypeptides or multiple subunits. And notice there you have 2 subunits. One is in a light gray, one's in a dark gray. Those are showing 2 different polypeptide chains. You're only going to have quaternary structure when there's interactions between multiple polypeptide chains or multiple polypeptide subunits. So let's talk about primary structure first. The primary structure of a protein is just its sequence of amino acids. And notice there, and we normally refer to amino acids by their number in their primary structure. So I can see that GLY, glycine, that is amino acid 1. And notice that says that has an amino end. So that's my N-terminus amino acid. If I go all the way around to, looks like amino acid 32, valine, that would have a C-terminus or a carboxyl group at the end. After primary structure, secondary structure is alpha helices, what you see on top. That is like the circular coils that form, but then also beta pleated sheets, and that kind of looks like a piece of paper. If you would fold a paper to make a fan, you could see that the folds are called beta pleated sheets. Now, those are all determined by the R groups of primary structure. So primary structure is the most important because that determines whether or not we will have just straight sections of the protein, or if it will make alpha helices or beta pleated sheets in its secondary structures. The last one that all proteins share is tertiary structure. Tertiary structure is also determined by the interactions between different R groups of amino acids. And there's going to be a few interactions that I'm going to highlight here. One is hydrogen bonds, and you can see that forming up in the upper left. There's also hydrophobic interactions. So you might have R groups that all consist of hydrophobic amino acids, and those are going to fold so that they are interacting with each other and not interacting with the aqueous or water environment. We also have ionic bonds that can form. That's going to happen between your acidic and your basic R groups on amino acids. And then the last thing that's kind of weird, we form disulfide bridges. So cysteine amino acids have sulfurs, and those sulfur molecules can form disulfide bridges, and that helps determine the tertiary or the 3D shape of a protein.